Uh, Thanks, Alison. Hi, everyone. Can you see my screen?
We can.
Okay, right. So thank you for the opportunity to present a little bit of the work that we as a team from the Pest Genomics team from CSIRO has been doing. I'll present a few examples of how genomics may be used to assist with managing of plant pests and diseases and how it may impact on our decision on how to develop IPM solution, and most important of all, how we can use it to strengthen biosecurity in the region and at a global level. So I want to thank a lot of let me find the pointer option. Laser pointer. Okay, I want to thank a lot of the funders. So this, for example, Grapevine, RDC, Research Development Corporation, Cotton, Grains, FMC's, Corteva, Dow, Sugar Research Australia, and also government departments such as Australian Foreign Affairs and Trade, DFAT, and also ACR. And also, most importantly, I want to thank Alison for organizing this webinar. So let me see. Now I can move. Okay. So the work will not be possible without the help of and contribution of the genomics team. So these are the current team members that are not in the red. We have John, Rahul, they are the sub-team leaders. But also I benefited from a lot of honorary and affiliates, so such as Alison and Carl and Darren. And I briefly counted there are about 24 people that had gone through the lab in the last few years. Many of them have contributed to helping to understand fall armyworms and Tutor Absoluta, which we are now embarking, led by Vinicius. And also, I'm going to present European Grapevine Moth today, which is led by the past team leader, member, Angel. So let me start my talk. So today I'm going to look at first section. I'm going to talk about molecular markers, how the decision to select a specific type of molecular markers will impact on our development of IPM strategies and ultimately plant health. The example I'm going to present is the coconut rhinoceros beetle. Some of you may have seen this, but I'm going to do a summary of it. So this is the beetle pest. It's quite a handsome beetle found across South Asia all the way to the Pacific, to countries bordering the Pacific, such as Philippines and Indonesia, but it's all the way across Southeast Asia. So this is a pest that is a serious menace to coconut palms. In the early 1900s, it was supposedly introduced from Sri Lanka to the Pacific, to Samoa and Fiji. And from there, it slowly spread through plant movements, and the population then exploded across the Pacific during the 1940s, during the military activities in that region. And in the 21st century, so recently, 2007, it was again detected in Guam. And what is important in Guam is that when this pest was found in Guam in 2007, a virus was researched to try to introduce to Guam to see whether it can be used to suppress this population. This virus is called the Orycters rhinoceros nudibvirus. It was first discovered in Malaysia in 1960s, and it was found to be very effective against the invasive population in the Pacific in the 1970s. It successfully suppressed the population such that coconut palms were able to grow and thrive. However, in the population that was discovered in Guam, the virus was found to be ineffective. So it appears that perhaps the Guam population has developed resistance. Molecular markers such as the mitochondrial DNA CO1 gene, widely used for barcoding, was first employed to determine whether it is the coconut rhinoceros beetles, and it was found to be the right species. Survey of the population then identified that various populations, such as from Guam, from Hawaii, Papua New Guinea, Solomon Islands, and so forth, all appeared to have the signature that is identical to the Guam population. It doesn't mean that it came from Guam. It just meant that potentially it came from Guam, or potentially these countries share the same origin as the population that came to Guam. More importantly, in recent years, between 2021 and 24, it's also detected in other countries, including the native range. So this is worrying because this Guam, or CIBG, G stands for Guam biotype or haplotype, is supposedly to have a really ferocious appetite for the coconut palm, and it destroys the palm really quickly. And there's no time for the palm to recover. So this is a really serious issue, and it's being addressed by international teams. Because of the identification of the CIBG, the Guam, an IPM decision tree has been developed involving development of new IPM solution. For example, if the beetle is found or palm damage is detected, one needs to confirm that the CIB is there, and then use the CO1 gene to determine the haplotype and test for the virus. If damage is continued to be seen, and the damage sign has been seen to be spreading, it can be assumed that the beetle has established. And the decision plan then goes into monitoring for plant damage, maintain sanitation, and importantly, biological control must be attempted. This involves releasing for the virus if it is not present. And most importantly, it is to try to match a virus strain that may be effective against the CIBG, the resistant one. This requires time and funding. However, when this report was developed, there is no effective strain of the virus being detected that could work on CIBG. So CIBG is not susceptible to the strain of the virus, and yet we need to find the virus that is able to control the CIBG. So while this is being developed and this is being researched, the beetle is killing the palm 24/7, ongoing every day, every year. So this is quite depressing. Now, in 2018, a group in Hawaii, University of Hawaii, looked at the population genetic structure based on the barcoding gene, as well as on nuclear polymorphic markers, just under 8,000 markers. What they were able to confirm was that the CIBG found in Guam is also identified in Hawaii, again in Taiwan, and also confirmed to be present in Palau, which Palau appeared to show to have two types, the Guam and the remaining of the, sorry, I forgot to mention this, the remaining of the other haplotype, which are susceptible, CRBS susceptible. Okay. And in the nuclear markers, they also identified that indeed the Guam population matching the colors, likewise Hawaii, Palau having the two types, which are here, and Taiwan having, again, unique population. But we must look past this initial analysis to really understand what this means. It means that in this Palau population, the Guam population, if they share the same signature, the population must somehow be admixed. And yet, when you look at the population here, Guam really just remain on Guam. Likewise, it didn't get into Hawaii, nor did it go into Palau or Taiwan. So having this G signature identified in this country, we should be seeing the same nucleus signatures in these countries as well, and yet we do not see that. It is saying that Guam population hasn't moved. So we are having the same populations having two different stories depending on the type of markers we're using. And this disagreement needs to be resolved. And it boils down to the type of markers that we're using. So the CO1 gene, partial gene, is here. And that represents around 3% of the mitochondrial DNA genome. And we're using 3% of the genome to try to distinguish between the Guam and the susceptible haplotype. This is a real data set, just looking at representative of individuals. Guam has this signature, and indeed it is shared with Philippines, with Rota, with Hawaii, Taiwan, with Papua New Guinea, Marshall Islands, and Solomon Islands. We will be assuming that anyone and any individuals here that is having this CRB, the Guam signature, would have the same signature across the rest of the genome. And yet when we look at it at the genomic level, and this is just looking at the mitochondrial DNA level, Guam is indeed different from the rest of the population. So the CO1, the region that is used for species identification when applied into understanding invasion biology, appear to be erroneous. We shouldn't be doing that. If we had chosen the other section of the CO1, we would be saying that every individual would have the same Guam signature. So having limited DNA region as marker can be quite dangerous when we are using it to infer invasion history. So if we look at the reassessment of the population, and we can see that in countries that are now reporting severe damage of the coconut palms, like in Vanuatu, in Solomon Islands, in Hawaii, and in Papua New Guinea, we would expect that the Guam populations should be there, or at least population having the Guam signature. At around 300 individuals of analysis at the full mitochondrial DNA genome, we see that the Guam population remain on Guam. More importantly, at the whole genome level, we can detect another Guam clade, and this represents a very recent new arrival. So this beetle is moving around, but we are unable to detect it using CO1. No other places have Guam otherwise. And that is now in agreement with the nuclear genome analysis. Importantly, we can see that some countries such as Palau is related to Indonesia, Hawaii is related to Philippines, sitting at the base of the clade, and Sri Lanka and Samoa are related, supporting the original anecdotal understanding of where the population from Samoa originated from, which is Sri Lanka. So now if we consider if Guam population hasn't spread, how do we actually reconcile between the observed severe damage of coconut palms and the supposedly failure to control using the virus? And that, I think, boils down to the fact that many of these countries were told that they have the Guam population or the Guam genotype, and these are resistant to the virus, and therefore introduction of the virus is probably not going to work, and they should be trying something else. And the virus is the most effective biological control agent, and they are not being released. And this is what is leading to the observed damage. And that's a hypothesis, and we should be testing that. And now we have the genomic resources, we should be able to test it to see whether this is being supported or not. So in terms of the introduction pathway, we can now look at historical proposed introduction pathway versus the current very complex pathway of the beetle movements across the Pacific. We have populations that are clustered as regional unique populations from the native ranges, and they're moving across the Mariana Islands and to Palau, and they are linked to historical link for over 100 years now between Sri Lanka and Samoa and Fiji supported, and importantly, Philippines potentially as the hotspot for many other places, invasive populations across the Philippines. So yes, we can identify biosecurity hotspots, Philippines and Taiwan here, being responsible for many populations in the invasive range, Indonesia to Palau, Philippines also to Palau. So we have two pathways into it. Malaysia associated with Papua New Guinea and Solomon Islands. Now, importantly, the rhinoceros beetle shares a native range with palm weevils, and these are really significant palm weevils, and is being detected across other invasive range. Are we looking at potentially the beetles using the same pathways to go into Indonesia? So this is going to biosecurity preparedness and helping us to start thinking about developing a toolbox to manage this palm weevil and to early detect the arrival of potential palm weevil populations. We can also use the genome to look at the biocontrol agent. So the Oryctys rhinoceros nudibranchs, we can characterize the genome. It's around 130,000 base pair long. We can look at Malaysian and Pacific Island populations being linked as historically being introduced from Malaysia into Pacific. Within the genome of these populations, they share a high level of genetic similarity, differ by only 0.5%. Likewise, if we look at the Indonesian isolate, they differ by about the same, but between Malaysia and Indonesia, the isolate differ by approximately 5.5%, so over 10%, 10-fold difference. So these are potentially different genetic isolates of the virus that's being described for the first time. And we can do that with a whole genome approach. And if we compare the base pair differences and the gene differences across the Malaysian and the Indonesian virus, we can then detect significant insertion or deletion in the gene, which is represented by the yellow, and hypothetical coding sequences that have huge deletion or insertion, indicating that these are unlikely to be genes, they're just intergenic regions. And these are the candidate gene regions that we potentially can look for differences that may lead to more effective management solutions. So this is some published data. So if you're taking screenshot or listening to it later, please consult me before you show it to other people. And finally, the need to match one strain of the specific virus to combat CRBG, is that the way that we should be moving? If we look at a real life scenario where we are looking at one CRB individual, we can detect many different virus isolates evidence being present. So in this case, we have, for example, a Malaysian isolate sequence here and a second isolate sequence here. Likewise, for Indonesian, we can determine that there's one batch of sequence that are identical to Indonesian that is represented by here, and the second batch, which has this region, but is highly similar to the Malaysian as well. So in the nature scenario, there is just not one specific strain of virus being present in a specific rhinoceros beetle. So why are we actually going down to try to match and look for one specific strain that might be effective? So this is something for us to think about, and potentially we should be evaluating the current management strategies. So now I'm going to talk about fall armyworms, and again, I've shown this before, but I'm just going to quickly take you through and looking at how we use genome to try to identify an alternative pathway for the fall armyworms. So as many of you know, that the fall armyworm was first reported in Western Africa in Benin region in 2016. And subsequently, with the detection date, it's assumed that it has spread from Western Africa to the whole of Africa, and then to South Asia, Southeast Asia, and so forth, and into the Pacific, and eventually to, for example, New Zealand. And these populations share the same, again, CO1 signature. So it seems like this is a founding population spreading very quickly. So why is it important to reassess the spread pattern of the fall armyworm? Because this is because current assumption is that it spread really quickly, and it's due to natural spread. They are very agile, they are very strong flyer, they can fly, and they take advantage of the wind, and they spread very quickly. The alternative hypothesis would be that it is actually assisted by human, and this is therefore trade-related. Because this is a trade-related activity, it involves behavior. If we change the behavior, we can reduce the risk. And this may be relevant for other spodopteran species or other invasive species yet to come. If we can increase and improve our phytosanitary behavior, we can potentially reduce and delay potential new introduction of invasive pests. So getting the introduction pathway right means that we can properly identify which might be the more plausible reason, and therefore develop a behavior and a response appropriately. So single introduction, I'm going to use this tutor absoluta example to demonstrate a point. So single introduction looking at nuclear sequences should ideally, should in effect be fairly homogeneous, because you have a single population, and the founding population, such as in the native population, should be the same as most of the subsequent introduced populations. So this is what we'll be expecting to see at the nuclear level. This, however, is based on microsatellite loci. So this is based on either 12 or 7, because there are 5 markers that are problematic, which I'm going to talk about a bit later. But for now, the purpose is to demonstrate that this is what single founder would look like. We have done a lot of mitochondrial DNA analysis. These are whole genome analysis, and we can demonstrate that between Africa and in the first studies that was published in Communications Biology, we can demonstrate that the mitochondrial DNA genome signature in, for example, China and India is actually quite different from, for example, Uganda and Benin. So this would suggest that there are multiple female founders rather than just a single female founder. And looking at the whole genome analysis, it is not the signature of a single founder. In fact, native regions are very different, and invaded region, again, is very different, and it's again different from what we see in China as well, suggesting that potentially we're looking at multiple introductions. Now, this work is a subsequent follow-up study led by Rahul, and he led the project and looked at the Southeast Asian populations. And again, we see very diverse signature. And we can see that, for example, in China, the population is very different. And this match another studies by Jiang et al. That look at within China populations, and even populations that are very similar, very close to each other, they're very different. In Malaysia, for example, we can see that there are at least three separate populations. And this suggests that these 4 armyworms potentially arrived multiple times in the region. And they did not match the signature of them spreading across from Africa, having the same signature into China or into Malaysia, and especially across into the Southeast Asian region. So if we look at the whole genome analysis, we can see that we identify hotspots in Malaysia, and in this time we identify in China. And there are populations that are moving very readily across between Southeast Asian countries. But also importantly, we are identifying unilateral movement, unidirectional movement from Asia into East Africa at the very least. So this match previous report that in Vietnam in 2008, there had already been 4 armyworm populations detected around Hanoi. And also in China, we were able to demonstrate that we had populations or individuals from 2016 intercepted and sequenced and confirmed that they were 4 armyworms. And this is in the years while the population was supposed to be in Western Africa. Now looking at the gene flow pattern, red represent highly structured populations, so they are not moving around, and green are populations that are moving around a bit. And these are pairwise comparison. And you can see that in the first year of Australia, populations are really quite unique. They are not moving around. And populations are sampled from Northern Australia, from Western Australia, Northern Territory, and Queensland, as well as New South Wales. And this population, by and large, they arrive as separate entity with very unique genetic signature. Whereas in Southeast Asia, Asia, and Africa, where the populations have been there for many more years, you can start to see that populations are starting to mingle. If we look at the population signature, we can see that Africa actually derived from Southeast Asia, East Asia, such as China or South Asia, India, neighboring countries, they're quite unique. And in Australia, we have populations that are dotted within Southeast Asia, indicating that they do have unique population signatures. And if we were to use a whole genome approach and just look at, for example, the organophosphate resistant allele frequencies, we can see that in Africa, the signature for F290V is quite unique between Western Africa and Eastern Africa. But more importantly, if we look at G227A, Africa population has no resistant allele. Likewise, when you move to into Asia and Southeast Asia, there's also no resistant allele, but all of a sudden, this resistant allele is detected in Indonesia. So there's many hundreds of alleles or thousands of alleles look at, and we do not detect this resistant allele, except all of a sudden it appeared in Indonesia. It didn't appear in Australia, but it somehow appeared in China. This signature suggests that these populations are likely to be independently founded. And in China is in Hebei, which is, I think is around this region. So if it were to be coming from Africa, it would move from west into east. So it's quite unique, again, supporting multiple introduction. So I'm going to look at now, very quickly look at the tutor absoluta work. So this work was led by a PhD student, Vinicius, who was in the team last year. And he surveyed literature plus doing some sequencing of African populations to demonstrate that in the current distribution of tutor absoluta, where we have full mitochondrial DNA genome, this pest has at least 8 introduction events. Are they specific to the region where they were sampled? We don't know because we don't have enough funding or time, this being a PhD study. But we did detect widespread insecticide resistance to pyrethroid. But what about to the others? So if you look at the current understanding of tutor absoluta diversity, where it is said to be single introduction, it is again based on CO1. And the gene for the CO1 region is by and large identical except for one individual in Kenya, this one, which has one unique substitution in this CO1 gene. So this is likely to be evidence of human-assisted introduction because there's very different mitochondrial DNA genotype. And we don't know how these populations are connected between the invasive population. And importantly, we actually do not have native populations as compared to the 4-1 populations where we have many native populations and many invasive populations. So this is work in progress, but we are setting the pace to demonstrate that, again, this very important tomato pest is actually likely to be multiple introduction. So how do we explain the example that I showed before, that based on microsatellite markers, it appeared to be single introduction? Well, I'm going to take you through a little bit of molecular evolution here. So microsatellite markers are fragment of DNA with repeat units such as TGA or CA or TC, and they repeated multiple times. And these repeat units, the similarity between the repeat units are indicative or is used to infer genealogy similarity. So if they're identical, they're more likely to share the same lineage. However, we know that from genome sequencing exercise and from extensive sequencing of the genome, microsatellite markers are likely to be related to transposable elements. And in this case, this is an example of association with transposable elements, which is called RTE. It has a TSD, which stands for target site duplication. I'll talk a bit more about that. Very quickly, though, we can demonstrate that RTE, this mobile element, here we have a full-length mobile element. It is found in the genome of Heliconius, a butterfly, and it has a repeat unit. So it has a microsatellite repeat unit and has target site duplication, where the insertion goes in there and is repeated at both ends of where the mobile element is inserted. So you have a 19 base sequence that are identical at the start and the end of the host genome where the mobile element is inserted. This is a signature of the mobile elements being moving around. But this type of mobile elements breaks up very easily. And when it breaks up, it's randomly insert itself into the genome. So in the genome of of a lot of Lepidoptera, you can see this partial gene of the mobile elements are inserted everywhere, and it create all sorts of microsatellite markers, repeat units that are then being isolated and being used to infer population movements and connectivity. So how is target site duplication occurring? So you have your host genome, and it is cut in a staggered way. So it cuts from here and then onto there, creating an overhang of the host genome. The mobile element inserts itself. And because this is DNA is double-stranded, we have a template here and here that are identical. The host gap repair mechanism duplicate this region and also duplicate this region to include to encase the mobile elements with the repeat units. And if you develop your markers here, you will get, for example, in this case, a heterozygous individual with a repeat unit of 7 or 8 CA. However, in some certain circumstances, one of the parents do not have this mobile element, and you are only amplifying this region from one parent, say, for example, from the mother, whereas the father didn't have this. You get no product, and you are generating a hemizygous band or individual, but you're interpreting it as a homozygous. All you can see is one band. Your interpretation is that it is a homozygous. Because they're homozygous, they're genetically more similar, therefore likely to be from the same origin. And if you look at the actual marker being used in tutor absoluta, you can see that these are the repeat units in yellow, These are the target site duplication, and these are potential mobile element fragments dotted everywhere. These are the poor marker that they sometimes excluded just to demonstrate, but also in the good markers, it is also present multiple times. And because of this, it can be a factor leading to the same, the interpretation that it came from the single founding population, and then the spread across the world. So knowing your marker's limitation is really, really important. And if we can, we should try to use a genomic approach to assess the spread and to identify potential factors. So finally, I'm going to talk about the European grapevine moth, and this as an example of how we can use genomic to prepare for the pest arrival. This is a pest of Palearctic region in the Mediterranean region. It was discovered in 2008 and 2009, being introduced to South and North America. Because the year was so close, it is not sure whether it actually arrived in South America and then went to US or whether it was the other way around. But Lobesia botrana, or European grapevine moth, is a really important pest to the Australian wine industry. It is one of the top 16 priority exotic pests that Australian wine and table grape industry is trying to keep out. This work was led by Angel, who was previously in my team. We're looking at populations representing the native region across the Mediterranean basin, from Western Europe to just bordering Asia, in Turkey, Syria, and also in Israel, as well as the population from Chile. We generated around 1,400 SNP markers, so genome markers, and we're going to compare that with a study by Reinecke from Germany, Renee's work, that look at 10 microsite loci, where they look at Spain, Italy, Germany, Turkey, and Israel, and Syria population as well. When we analyzed the populations, we saw that the population split into 4. Importantly, we see that the native population match pretty much the geographic region of the native range, such as France being by itself, Spain and Portugal being by themselves, being neighboring to each other, Italy, Switzerland, and Germany being very similar, as you know, the map of Europe, and also Turkey having a unique population, Syria and Israel are very similar. But we can also identify that Chile population was very similar to France, whereas the US population in gray were within the cluster that has Italy, Switzerland, and Germany. And we identified that the best way to characterize the population was 3 genetic cluster, as opposed to the microsite loci that only identified 2 genetic cluster. Now, Lars Germain, an expert in molecular evolution, then look at the phylogenetic pattern of this invasive and native population. And we can show that the population in US, in fact, is matching to Italy, and in Chile is matching to France. So we can identify the native range and the invasive populations. But importantly, we also identify movements within native range, such as Italy going into Turkey. Again, Italy seems to be repeating itself. And France, again, being detected in Turkey. Again, France is detected. So these are identified potential biosecurity hotspots, but importantly, allow us to understand that this species, despite the fact that they are winged insect, they are quite sedentary. They don't move a lot, very much. So if this species should arrive in Australia, we can actually develop boundary, because if we can quarantine it and we can set up the boundary, we can have a good chance that we can localize the invasive population and stop it from moving to other regions of Australia. So in summary, market choices are very important. They can impact on IPM strategies and ultimately impact on plant health. And we can look at the example from the rhinoceros beetle. Do we really need to match the virus with the mitochondrial DNA haplotypes, or do we introduce as wide a genetic diversity as possible and let nature do its selection for us? Understanding the limitation of the marker is very important. So I've demonstrated the efficacies of CO1 as species identification, and we should refrain from using it to infer invasion history because often they're not. Using a single small piece of DNA, we might get it wrong. Microcellulose, yes, it has better power, but we need to understand what factor can affect the marker proficiencies. Whole genome sequencing, I haven't had a chance to talk about it, but best examples are seen in the contrasting studies where we have studies that I'm presenting where we say that it's multiple introduction versus other whole genome studies where there's other research group are saying that fall armyworm actually came from Africa. So we need to, once again, go into look at the whole genome sequence approach and analysis method to really understand what caused the different in interpretation. Perhaps that can be another time. We can have better understanding and clarity of introduction pathway and the factors. And this is seen in tutor absoluta, where we can see that really we think that it is human assisted with multiple mitochondrial DNA genome. And then that this needs to then lead to a change in behavior and also a change in phytosanitary practices. Finally, for the Lobesia botrana, this is an example of how we can use the whole genome data to better understand the population movements and how likely they are to move, and then to use it to potentially develop future quarantine policies and set boundary limits. So with this, I'm going to stop my talk, and next you're going to see genomic resources to help us understand novel solutions and to manage and impact these invasive populations. There are too many people to thank, and many collaborators and many countries. List them all here, but I'm not going to mention them all. I'm 2 minutes over the time, and I apologize.
Tek, that was wonderful. No, perfect timing and a great presentation full of information. If you've got questions for Tek, which I'm sure you have, please put them in the Q&A. We've got a few questions there, but I'm sure you've got many, and I actually do have quite a few questions as well. Just quickly, I'm going to ask you too before we go into B's presentation. And B, if you want to load your presentation up and get it ready, and while you do that, Tek, I mean, you pretty much you well set out the importance of looking at using a genomic level whole genome approach, rather than just looking at very limited sort of DNA regions. Why do you think people aren't doing that more? Because as you gave us some examples, people are still looking at sort of small parts and not taking a whole genome approach, and then they're missing really important key points and how to identify and manage different populations. Why are people not moving faster? Is it the cost? Is it the time? Is it the skills? Is it a combination?
All the above and none of the above. In many ways, the cost is no longer an issue. It has always been perceived as doing whole genome sequencing is really expensive, but that's no longer the case. In fact, if you do a single PCR of of the CO1 genes, and you're relying on a single marker, you may have to repeat the PCR and sequencing multiple times before you get the good result. And you're spending almost as much, if not more, for whole genome sequencing of an individual just to get a tiny piece of DNA. Technical issue and expertise, that will be, I think that is currently a very big hurdle for many regions, but we're improving and we're developing a platform to support this. So don't give up. We're doing our best to try to push out the solution for everyone.
Excellent, thanks, Tek. And another question, I mean, when you look at populations at genomic level and you see these differences in populations between regions, which is quite clear on a lot of these diagrams, And you gave an example with CRB around how there's a virus that works for one type and it doesn't work for the other. But in general, I mean, is there that much difference between, you know, the efficacy of biocontrol solutions for different populations? I mean,
Yeah,
oh,
look,
Auto.
Alex, I'm glad you mentioned that because after I moved away from the CRB slides, I realized I forgot to mention. So the CRBG, the perception that it is resistant to the virus, we actually do not know why that is the case. But if you read the original reports for associated with the experiment that did the bioassay challenge, the experiment was facing a lot of difficulties, especially scoring the mortality rates because there were a lot of contamination from metarhizium, for example, where the larvae were dying and it could not be clearly attributed to either metarhizium or the virus. And so it requires repeating. And importantly, we actually never saw whether the larvae on Guam were challenged side by side with, for example, a native population. So we can't really say that they have elevated resistance or tolerance because we don't really know what is meant by a resistance. It could very well be the fact that the virus is infective because there is a mutation in the virus genome, or perhaps it is truly resistant, but we still need to be able to confirm with better controls. So can't answer that. I'm looking for funding. If anyone in here, in this audience, has
Nice try,
and
Tek.
spare funding,
So
please let me know. We can collaborate and get to the bottom of it.
excellent. So I've got another just a quick question, and I've got more to talk about in the panel discussion. But we did have a quick comment here that Vanuatu was not mentioned in the spread of CRB, and it has both strains. And then there was sort of apologies. Look, I saw it now mentioned, but just to clarify, what you're saying is that the CRB Guam type has not moved out of Guam, according to what you've seen. So therefore, is it in Vanuatu or is it not in Vanuatu, according to what you've seen?
I was in the process of answering that, and I will answer it here, but I will also answer it later. So Vanuatu, we actually have no whole genome analysis because this work for the coconut rhinoceros beetle was conducted during COVID time, and it was very difficult to get material. We were very lucky to be getting so many populations from many different countries, but we missed Vanuatu. So I can't say that Vanuatu truly hasn't got Guam because we haven't got the data to support that. But based on everything that we have so far, there is no evidence that Guam population or the population that went into Guam actually gone anywhere else.
Okay, excellent. Thanks, Tek. So a couple of questions. I guess, Heng, you talked about lots of those challenges, complexity of the data, the volume of data, methodological challenges. Do you think AI, therefore, is going to be quite a powerful tool to sort of maybe help us fast track, be more efficient in managing all that data and actually focusing our minds on some like key aspects a lot quicker?
Yes, I think AI is I mean, it's like any tool is if you use properly, it can become really useful. And but I guess on the flip side, we need to be really careful about using it. So like you still need to have a level of skepticism about what it comes out. And so you need to have ways to make sure that it's actually doing the right thing. So I guess in that sense, yeah, so with a grain, yeah, a little bit of skepticism is good to have, but it definitely, if used in the correct way, it probably will help research a lot. Like, I mean, from my personal opinion, I just using a chatbot, it helped speed up writing some of my codes a lot more. Like some things that would have taken me maybe a day just to figure out, as just within a few seconds. I was like, oh, this is really good. And then I can test like, oh, it works. And it's like, okay, yeah, then it makes sense. So yeah. So that's one thing I can testify.
Excellent, thanks. I can see B is nodding her head as well, and I'm sure everyone's looking and using AI. We're going to have a session on AI and biosecurity pest management control later on in May, and Rahul's going to be there for that. Rahul, I know that you're looking at that though as well, around how to use AI to sort of maybe, I don't know, bring these sorts of tools as well and these sorts of I guess solutions to a wider audience as well, because I agree, and you have to be skeptical. You have to know what you're doing still, because you have to know where it's not telling you the right thing. But it also does open up a huge opportunities for people to access some of these technologies that they've never been able to before. Rahul, you're working on some of that work now. Is that true?
Yes. So I think, I mean, as Heng mentioned, right, one of the things that we have been quite interested in for the last 2 years is making sure that the decision makers have the right information at their fingertips when they need it, rather than after the fact. And we want to avoid the next fall armyworm or the helicobacter pylori situation. The approach that we've been taking is, our team and a lot of our collaborators have been doing this for years, for decades. And so there's a lot of information that is already there, which we can actually use to help what we call evaluate and benchmark AI solutions that we are building. And so we've sort of in a fortunate situation where we do have a lot of data that we've generated. We do have a lot of analyses that we've done, inference that we've delivered. And so we can actually use that to what we call guardrail the AI solution, so that when it is used, it is going to deliver information, and only that information that it is confident in, and nothing else. And so that involves a lot of manual effort to actually do that. But I'm slightly more optimistic than Heng. That's probably an affliction of mine. But we are very quickly getting to a point where the boring stuff, at least, is likely to get automated confidently. But the important things like the inference, that's what the scientists or the people on the ground need to deliver. And so all our focus is going to be on getting information to the point where it can be inferred, and let's do that in 6 days and not 6 months. That's basically the running ethos we're operating under.
Yeah, thanks, Rahul. And Tek, a question for you around this. I mean, not just with AI, but with all these tools that we're using, it does rely on good data that's gone in. And Heng talked about the reference genome and having like a library and stuff. But if the information in the library isn't great, how do we fix that? So when we're using some of these tools, and we've been in workshops, I've been in workshops that you've delivered, and we've seen some sort of not always robust references in the library. How do we go about cleaning all that up and making sure that that data that we rely on for decision making and the tools rely on and draw on, including AI, is more robust?
Well, we can try to rely on AI in the future to try to help us to recognize that. But I guess there are two solutions, somewhat selfishly. One, you get a really good team like Rahul and Heng and Beatrice, who are really good with their genomics, and get them to do it. Collaboration with for example, with Professor Yidong's group involving Jianpeng, we really achieved a lot. The other one, I think, to recognize and fix it, it really comes with a lot of experience and learning from collaborators. I don't think there's an easy solution. It does come with experience, recognizing that like this is probably what it shouldn't look like. Trusting your gut feeling and be there to step outside the box and challenge it, I think. So
Excellent.
testing.
I like to hear that, because that's science. We should be testing and questioning what we find, and we should be evolving our thinking, particularly as we get more insight through more powerful technologies and research approaches. So I think that's a great message. You've got lots of thumbs up there as well. I guess a question maybe for all of you as well. Maybe we'll start off with Tek, and then I'm going to ask B as well. But you kind of showed really that Southeast Asia, for example, is a real sort of biosecurity hotspot. I guess that's what a lot of your research is pointing to, and it seems like it doesn't really matter which insect it is or pest. It is, there's something happening there. So what do you think kind of needs to happen more in that space?
Yeah, good question, Alison. Yes, indeed, we did identify that Southeast Asia had been the vulnerability in terms of helping to or preventing the arrival of new pests seems to be facing a bit of a challenge. But it reflects somewhat, the fact that Australia is an island, so we can't really compare that way. That said, in Australia, we actually have a lot of problem as well, and we have contributed to global pests as well. For example, brown tree snakes eating up all the endemic bird species on Pacific Islands, for example. With from Australia, the eucalyptus borer beetles, borer cancer has spread around the world. So it depends on the pest system. In this case, the more recent significant pests somewhat pointed to Southeast Asia and Asia as a whole. I think to overcome that, it really requires the government and researchers to collaborate. There's a will to want to improve because ultimately you're only as strong as your next link. So we all can be weakest link, and this is probably an opportunity to learn from each other and help each other, because ultimately we're helping ourselves.
Excellent. And actually, I'm going to ask you another question because I just remembered something I did want to ask you. But Heng, you've got a question as well in the Q&As. Maybe you can have a quick look at that because I think it's quite relevant to your talk. Tek, somebody asked about the different strains of, I think, coconut rhinoceros beetle in the Q&As. And I guess I hear this quite a lot, different strains, fall armyworm as well, rice and maize strain and stuff. And what's your sort of thoughts on this? Because I think you were sort of saying in your answers on the Q&A was maybe we should not be thinking about it so much sometimes as strains or feedback.
Yeah. I think that in many ways, what we name something tends to lead to a misconception or premature conclusion. So, for example, the white flies, Bemisia tabaci, which I've worked a lot of, again, the naming of the complex or the species tend to lead to very different management strategies. For this purpose, for the rhinoceros beetles, the naming of the strain, so to speak, the guam or the susceptible, is very, in many ways, misleading, because when we talk about guam, people would tend to assume that it all came from Guam, but Guam actually got it from somewhere else. So it's just only, like what it has been first identified in Guam and subsequently the same partial signature identified elsewhere. But the genomic analysis doesn't support that, other places having Guam. So it doesn't help to keep calling it Guam or S, susceptible. For Spodoptera fujiperta, it's a bit different. So when we talk about Jane Peng's study on Helicoverpa armigera, I mentioned about the subspecies genomic status being, between Australia and armigera. Armigera is around 100,000 years, but however, between armigera and the Xinjiang population is about 200,000 years. So is it a subspecies or not? Well, probably, but what about Zea? Zea differ from armigera by 1.5 million years. Okay, we call it two different names, Helicoverpa Zea and Helicoverpa armigera, because of 1.5 million years. But if you look at Spodoptera fujiperta, it's over 2 million years. We call it a strain.
Yeah.
So the naming of the species ultimately lead to a lot of decisions, and it's very difficult. Evolution is not punctuated, it's probably continuous. So yeah, it's very difficult to say what we should be calling it, but we should be very careful. And I think having more genomic resources as what we are now demonstrating is going to help.
Great, thanks, Tek. Great answer. When you were doing your research, I mean, you've looked at some biocontrol options there within Australia. Did you sort of look at what was being done across the greater region? And is there anything there that you're seeing that is of interest as well in other countries?
You mean like outside Australia in
Yeah,
general?
outside Australia, yeah.
Yeah. So I think there are a lot of research being done in the field of entomopathogenic fungi in particular, and even within Australia, I think. And there are a lot of commercialized fungi that out there, especially in US in particular, or in that region, because four mealworm was native there. I'm not quite sure about other country, but yeah, I assume that might be some already outside Australia.
Yeah, excellent. Thanks, Faye. And good luck on. Have you actually finished your PhD? Can I say congratulations yet, or are you still?
Yes, I finished. Just waiting for the confirmation later. So I think yes, it's finished.
Okay,
Yes.
I'm going to say congratulations then on behalf of
Thank
everyone,
you.
because that's a big effort and it's always a big thing to do. So it's congratulations from me. Rahul, you were really involved in the project around in Southeast Asia with Tek and fellow colleagues. And I guess thinking about that, where do you sort of see kind of the opportunities for genomics from your perspective in the future, near future, let's say in the next 3 years?
So genomics is highly mystified, right? People think this is a very shiny, expensive thing. It's actually not anymore. It's a lot more accessible than it was even 1 year ago, right? The cost goes down really rapidly. What's important now is not the technologies to develop to generate the data that can be accessed. What is important is the local insights. I think being able to share local insights and observations, anecdotal observations, like when Tek was going all around Southeast Asia, trying to identify what was actually happening. Anecdotal observations were quite important, saying in this country this was happening, in that country that was happening. But being able to talk to each other, I think Southeast Asia is one of those hotspots, societal hotspots as well, where people actually talk to each other a lot more than they actually realize. And so it's a much easier to actually collaborate within Southeast Asia than it is to collaborate in most of the world, even Europe sometimes. And so I think that opportunity that's a big opportunity for Southeast Asia that I think should be leveraged. The technologies are going to exist, and one of our primary focuses has been on making sure that we can bring those technologies to collaborations with us and make it much more accessible. But if that collaboration across those communities can be harnessed, I think that's where the actual power is. It's incredible, really, the way Southeast Asia functions.
Well, thanks, Rahul. That was a great way to actually end it. I'm just going to use that as my final statement, actually. That's perfect. And I'm going to second that third and fourth, fifth, whatever, because Southeast Asia and the region and working with Australia is just a really proactive group of people right across the region that are really ready to share and work together proactively. And I think, Hen, you mentioned the importance of sort of that future collaboration really well in your, in fact, you all did in all the presentations. So this is a really good example today, and I look forward to continuing. Thank you so much for joining us, to all the speakers. Fantastic presentations, really, really interesting. We only just touched the iceberg. Sort of, there's so much more to talk about. Our next session in May, we actually have a bioassay step-by-step masterclass in 2 weeks' time, which will be excellent if you're really interested in conducting bioassays. It's going to go through really each step. And that will be really, really good. And I think Valen and a few other speakers that will be in that session actually joined us today. And Rahul's going to come back in May, and we're going to talk about AI as well and the potential for that, which will be really interesting because that's very exciting as well to all of us. So thank you so much. And we will see you. I will share the presentations. I'm sorry we went slightly over time, but that happens with such good speakers and such good presentations. Have a lovely day or night or morning, wherever you are joining us from, and we'll see you next time. And thanks for all the emojis, guys. That's awesome. Okay,
Thank you.
bye.
Thank you.